So let's continue with small molecule neurotransmitters. Now, these neurotransmitters are synthesized in certain neuron cells, and pay attention to the details in here. Well, the enzymes that are responsible for synthesizing small molecule neurotransmitters are synthesized in the endoplasmic reticulum. So it's the enzymes, not the neurotransmitter. So the enzymes are synthesized in the endoplasmic reticulum, and then they are transported into the Golgi apparatus where they are modified and so on. And then you have transport of these enzymes either in vesicles or soluble enzymes. They are transported into the synaptic terminal. Now, in the synaptic terminal, you have the synthesis of the neurotransmitter itself. So the precursor of these neurotransmitters is taken up into the cells via certain carriers or transporters, and they have synthesis of the neurotransmitters in the cytosol. And then they are packaged into synaptic vesicles, where they can be modified further. And once they're sitting inside these vesicles, the vesicles wait for a signal which is terminated. It ends with influx of calcium ions, and that promotes fusion of these small synaptic vesicles into the plasma membrane, and the neurotransmitter is released. Now, once it's outside, it can bind to receptors on the postsynaptic cell membrane, inducing a certain signal, and it can be terminated either by diffusion, so that it goes away from the site of release, or it can be taken up by the presynaptic cell, or it can be enzymatically inactivated outside of the cell. Now, as I mentioned before, release or fusion of the large dense core vesicles that contain the neuropeptides require influx of large concentration of calcium ions, and this influx, the site of influx can be far away from where the site of fusion of these vesicles. On the other hand, with the small synaptic vesicles, the fusion of these vesicles with the plasma membrane occurs close to the site of entry of calcium ions, and that just shows the concentration of calcium, the amount of increase of calcium ions inside cells once they get into the cells. So these are nice electron images of small synaptic vesicles. You can see how many there are inside these cells. Compare the size of the small vesicles with the large dense core vesicles right here. Now here you have a fusion of the vesicles with the plasma membrane, and here you have endocytosis. That is, you have these vesicles that take up some of the neurotransmitter back into these presynaptic cell. So what is the factor, or what does determine whether these vesicles fuse with the plasma membrane or not? I mean, consider the large number of vesicles that are just sitting in here and waiting for the signal. So the signal is the influx of calcium. The importance of calcium is that it stems from the fact that what it does is that it binds to a protein that is located on the surface of these vesicles. The protein's name is synaptotagmin. And once calcium increases inside these cells, it binds to synaptotagmin. That allows synaptotagmin to interact with proteins that exist on the plasma membrane. And that puts the vesicle very close with the plasma membrane, allowing for the fusion of these vesicles with the plasma membrane and release of their content. So let's talk about some examples of small molecule neurotransmitters now. We'll start with the tyrosine-derived neurotransmitters, also known, we'll start with the catecholamines that include dopamine, norepinephrine, and epinephrine. Now I want you to pay attention to the cofactors that are needed for the synthesis of the small molecule neurotransmitters. Specifically, the S-adenosylmethionine, which is important for methyl transfer, pyridoxal phosphate, which is important for transamination and decarboxylation reactions. Pyridoxal phosphate is derived from vitamin B6, and tetrahydrobiopterin. So we'll start with the catecholamines. Now, tyrosine is present in food products, and it can be synthesized from phenylalanine. So it is a hydroxylated phenylalanine. So tyrosine comes from diet or from liver, and and tyrosine can be converted into DOPA. This is the first reaction, and this reaction, in fact, is the rate-limiting step, because this enzyme is highly regulated, and we'll talk about its regulation. So I have the formation of DOPA, and it's a by hydroxylase enzyme. So this is the hydroxyl group that is added. This reaction requires tetrahydrobiopterin as a cofactor. Now, these are collectively known as catecholamines because this is the structure of the catechol moiety. Now, DOPA is converted into dopamine by a decarboxylase enzyme, and this enzyme requires pyridoxal phosphate as a cofactor. Now, DOPA right here, so you have decarboxylation, so removal of this CO2 or this carboxyl group, and for forming dopamine. Dopamine is converted into norepinephrine vesicularly inside vesicles by hydroxylase enzyme. So you have the addition of a hydroxyl group right here, and then you have the addition of a methyl group to norepinephrine, forming epinephrine by a methyltransferase enzyme. Now this enzyme requires S-adenosylmethionine as a cofactor. Okay. So let's talk about dopamine now. What happens is that once dopamine is synthesized in the cytosol, it's packaged into the small synaptic vesicles. Then you have a signal, calcium influx, release of dopamine into the synapse. Now, most of this dopamine, well, some of it, of course, will bind to receptors on the postsynaptic cell. Most of the dopamine is taken up again by the presynaptic cell, and 50% of this dopamine is repackaged into synaptic vesicles, and some of it goes into the mitochondria, where it can be inactivated by monoamine oxidase enzyme. Now, some of the dopamine diffuses away, and it can reach the liver, where it can be inactivated by monoamine oxidase or the catechol or methyltransferase enzyme. Now, about 10% of dopamine can be taken up by the postsynaptic cell, where it can be activated by the methyltransferase enzyme. Now, an important point to mention here is that the reuptake of dopamine into the presynaptic cell can be inactivated by cocaine or amphetamine. So what these do is that they keep dopamine for a longer time in the synapse, resulting in amplified signal. Now, in terms of norepinephrine, dopamine is packaged into vesicles, which are converted into large, dense core vesicles. Now, inside these vesicles, you have the enzymes that are required for converting, the enzyme that is required for converting decarboxylase enzyme that is required for converting dopamine into norepinephrine. And then norepinephrine can be is packaged into these vesicles that fuse with the plasma membrane and released. Again, just like dopamine, most of it is taken up by the presynaptic cell, where it can be repackaged or it can be inactivated by monoamine oxidase. Some diffuse away, and they reach the liver, where it can be inactivated. But doesn't seem that norepinephrine is taken up by the postsynaptic cell. Again, the transporter that is needed for reuptake of norepinephrine can be inactivated by amphetamine. As for the synthesis of epinephrine by the methyltransferase that converts norepinephrine into epinephrine. Now, as I said, norepinephrine is now inside vesicles, but it leaks out of these vesicles. So norepinephrine becomes cytosolic, and it can be converted into epinephrine, and epinephrine is packaged into the small synaptic vesicles. Okay. Again, same thing happens. Epinephrine can be taken, once released, it can be taken up by the presynaptic cell, or it can reach the liver where it can be inactivated. So how are these the catecholamines, specifically dopamine and epinephrine, packaged into these synaptic vesicles? Well, what's needed here is a proton pump, an exchanger. It's a pump that exchanges the monoamines with protons. Okay. Now, so you have to have high concentration of protons inside these vesicles to allow monoamines to get into the vesicles, and this is done by a transporter known as vesicular monoamine transporter that pumps protons inside these vesicles. Okay, and it's ATP dependent. Now, dopamine, once it's released, can be inactivated, just like epinephrine and norepinephrine, but dopamine can be inactivated by monoamine oxidase and the catechol or methyltransferase enzymes sequentially. So you can start with the monoamine oxidase followed by the methyltransferase, or methyltransferase and then the monoamine oxidase. The final product is known as homovinylic acid. Now, this inactivation requires S-adenosine methionine, vitamin B12, and folic acid. Now, one point here is that homovinylic acid is a marker for Parkinson's disease. So it turns out that the ratio of homovinylic acid to dopamine is high in patients with Parkinson's disease. I should let you pay attention to a mistake. It seems to be a mistake in Marx, where it says that homovinylic acid is actually decreased in patients of Parkinson's disease, but I looked into different research articles, and it seems that it actually increases. So the rate limiting step is catalyzed by the enzyme tyrosine hydroxylase, and it's highly regulated. And it's regulated either short-term or long-term. Now, short-term regulation of tyrosine hydroxylase occurs by the end product, the cytosolic catecholamines, the dopamine, epinephrine, and norepinephrine. So high concentration of these catecholamines would compete with the tetrahydrobiopterin binding to the enzyme, so inhibiting the enzyme. Now, if there is prolonged activation, or if there is extensive depolarization of the neuron, that would result in activation of several protein kinases, such as protein kinase C, protein kinase A, calcium calmodulin dependent kinases, and these phosphorylate tyrosine hydroxylase. The phosphorylated enzyme can bind more tightly with tetrahydrobiopterin, and it becomes less sensitive to the catecholamines. Now, the tyrosine hydroxylase enzyme can also be regulated at a long term, and the same regulation mechanism occurs for dopamine hydroxylase, where prolonged sympathetic neuronal activity increases the amount of or the transcription of the tyrosine hydroxylase and the dopamine hydroxylase mRNAs. So these are the catecholamines. Let's continue with the tryptophan-derived neurotransmitters, and there are two of them, serotonin and melatonin. Now, the synthesis of these two enzymes takes place, or the precursor of these two hormones, the precursor is tryptophan. Now, the first reaction is the rate-limiting step, catalyzed by tryptophan hydroxylase. It's the same concept as hydroxylation of tyrosine. So this reaction requires tetrahydrobiopterin. Now you have the formation of the 5-hydroxytryptophan, which is decarboxylated, forming serotonin or the 5-hydroxytryptamine. Serotonin is packaged into vesicles via the same mechanism as the catecholamines, the vesicular monoamine oxidase monoamine transporter. Okay, and then it gets released once the neuron is activated. Now, many of the serotonin neurotransmitter is taken up by the presynaptic cell, and some is some diffuse away into the liver, where it can be modified further into a product known as 5-hydroxyindole acetic acid, which can be excreted in the urine. Now, the reuptake mechanism of serotonin is a target of many of the antidepressants. So these are collectively known as selective serotonin reuptake inhibitors. So serotonin stays for a longer time in the synapse, and that sort of like lifts up the mood of depressed individuals. Now, melatonin is in the pineal gland can be synthesized from serotonin. So what happens here is that tryptophan is converted into serotonin, and serotonin is converted into melatonin in the pineal gland. And this hormone is important for sleep patterns, seasonal circadian rhythms, and dark light cycles. So people who travel long distance, like for example to the US, from Jordan to the US, would take melatonin in order to fix their sleeping pattern. And it turns out that infants do have high level of melatonin, and that's what makes them sleep for a longer time. I know my son didn't have a lot of melatonin. Elderly people tend to release less melatonin, and that's what makes them stay awake for a longer time or sleep for shorter periods of time. So let's continue with glutamate and aspartate. Now, these two amino acids are non-essential amino acids. They do not cross the blood-brain barrier, and they have to be synthesized in the neurons. They can be synthesized in the neurons themselves or glial cells, and both are excitatory neurotransmitters. Now, in terms of glutamate, there are different sources for glutamate. It can be produced from alpha-ketoglutarate, which is an intermediate in the Krebs cycle. So it can be converted into glutamate. Or glutamate can also be produced from deamination of glutamine, removal of an amino group from glutamine, resulting in the formation of glutamate. And glutamate can also be synthesized from aspartate via transamination reaction. Once released, it can be taken up by the same cell, by the presynaptic cell, via a transporter known as the excitatory amino acid carrier 1, or it can be taken up by the neighboring glial cell or cells via transporters such as glutamate transporter or glutamate aspartate transporter. Now, once inside the glial cells, it can be converted back into glutamine. Glutamine is released, it goes into neurons, and again the cycle is renewed. Glutamine is converted to glutamate. So this is that shows the different reactions for converting glutamine to glutamate and to alpha-ketoglutarate and vice versa. Now, aspartate, there's some talk about aspartate not being a neurotransmitter since it hasn't been found to be vesicular. Since it hasn't been found to be taken up again by the presynaptic cell. So there's some talk about it not being a neurotransmitter, but it can be synthesized from oxaloacetate as well as glutamate by a transamination reaction. Glycine is also considered a neurotransmitter, and it is an inhibitory neurotransmitter. It is synthesized from serine, and serine comes from 3-phosphoglycerate. So you have glucose, glycolysis, 3-phosphoglycerate that is converted to serine, and serine can be converted to glycine. Folic acid is required for the synthesis of glycine from serine. Okay. And then we have the gamma aminobutyric acid, another neurotransmitter that is present in high concentrations in many brain regions compared to other neurotransmitters such as the monoamine neurotransmitters. And GABA it goes through what is known as a GABA shunt, and the GABA shunt, the main purpose of the GABA shunt is to produce GABA or to conserve the supply of GABA, considering that it's needed at high concentrations. So this is how GABA is synthesized. Again, glutamine is converted to glutamate by glutaminase. Glutamate is converted to GABA by an enzyme known as glutamate decarboxylase. Again, this is a decarboxylation reaction. It requires vitamin B6 or pyridoxal phosphate. Now GABA is stored in vesicles and it is released. Now GABA is either taken up by the presynaptic cell or it goes through the GABA shunt in glial cells. So in glial cells, what happens is that GABA goes in, it gets converted to glutamate. Glutamate is converted to glutamine. Glutamine is released out of glial cells and it goes into the presynaptic cell or another neuron where it can be converted to glutamate and it's packaged in small synaptic vesicles and it's released again. So this is the GABA shunt. So another small molecule neurotransmitter is acetylcholine, and acetylcholine is synthesized from phosphatidylcholine, which exists in the plasma membrane, or choline can be obtained from diet. So once inside neurons, it's conjugated with acetyl coenzyme A, resulting in the formation of acetylcholine. Again, packaged in inside synaptic vesicles, and it fuses with the plasma membrane when the neuron is excited, and acetylcholine is released where it can bind to receptors on the postsynaptic cell. Now, acetylcholine is inactivated by an enzyme known as acetylcholinesterase into acetate and choline. Again, choline can be taken up by different cells, neuron cells, and they can reform acetylcholine. Histamine, another small molecule neurotransmitter, it does not penetrate the blood-brain barrier. It has to be synthesized in the brain, and once so it can be synthesized from histidine by the enzyme histidine decarboxylase. Again, it's a decarboxylase enzyme. It requires pyridoxal phosphate. So you have the formation of histamine, which can then be packaged into vesicles via the same mechanism as the monoamine neurotransmitters. And once released, it gets inactivated by the histamine methyltransferase and an enzyme that is similar to the monoamine oxidase, but it's known as diamine oxidase or histaminase. Now, an important point here is that this inactivation takes place in astrocytes, and there is no reuptake mechanism for histidine back into neurons. So pay attention to that difference in here. Okay, so finally we'll talk about nitric oxide. Nitric oxide is a gas. Its half-life is very, very short. It's about 2 to 4 seconds. And it can be inhibited by hemoglobin as well as other heme-containing proteins. Now, nitric oxide really makes a lot of exceptions in terms of the definition of neurotransmitters. First of all, it is synthesized from glutamate. So glutamate can act on certain receptors located in the postsynaptic neuron. Okay, so what happens here is, so you have a release of glutamate. Glutamate binds to these receptors on the postsynaptic neuron, and that results in entry of calcium ions into these cells, as shown in here. This calcium ion would activate nitric oxide synthase enzyme, and this enzyme is important for converting arginine into citrulline and nitric oxide. Now nitric oxide is synthesized in the postsynaptic cell. Now nitric oxide is then released, and it can diffuse and enter neighboring cells, including the presynaptic cell. So not only neighboring neurons, but also the presynaptic cell. Now, once it goes into the cells, you have the production of cyclic GMP, and cyclic GMP will then bind to different channel proteins and different proteins, activating them. Okay, so the fact that nitric oxide can diffuse out of the postsynaptic cell and it goes back into the presynaptic cell, this is known as the retrograde. So nitric oxide is known as a retrograde messenger. Okay, so nitric oxide, what it does is that it activates guanylate cyclase in different cells, including the postsynaptic cell itself, resulting in the formation of cyclic GMP. So really, the question is nitric oxide a neurotransmitter? According to the definition, no. But it is considered a neurotransmitter, even though it is not stored in vesicles, it is not released by calcium-dependent exocytosis, rather it diffuses, its inactivation is passive, it decays spontaneously, it doesn't interact with receptors on target cells, its sphere of action depends on the extent to which it diffuses, and its action is not confined to the conventional presynaptic or postsynaptic direction. Nitric oxide acts as a retrograde messenger. It regulates the function of axon terminals. Anyhow, so according to definition, it's not, but in fact it is. There are 3 enzymes that are important for the synthesis of nitric oxide, and these are collectively known as nitric oxide synthase, and there are 3 isoforms, 1, 2, and 3. They are expressed in different tissues. Specifically, isoform 1 is important for synthesis of nitric oxide in neurons. Okay. Now, all 3 forms require dihydrobiopterin as a cofactor, as well as nicotinamide NADPH as a coenzyme. So, we're getting to the conclusion of this lecture. I want you to know the differences between between neuropeptides and neurotransmitters in terms of activity. The activity of small molecule neurotransmitters is fast. Neuropeptides, it is slow. In terms of response, again, slow in neuropeptides, fast for neurotransmitters. The duration of action, it's long for neuropeptides but short for small molecule neurotransmitters. There are different receptor targets for neuropeptides, but specific unique targets, receptor targets for small molecule neurotransmitters. Neuropeptides can affect gene expression, but not small molecule neurotransmitters. There are differences in terms of synthesis, transport, packaging. So we talked about that before. The concentration of neuropeptides that reach target cells is low, but it is high for small molecule neurotransmitters. Speed of release is slow for neuropeptides and fast for small molecule neurotransmitters. There are differences in terms of the concentration of calcium ions required for fusion of vesicles containing neuropeptides versus small molecule neurotransmitters. There are differences in terms of site of synthesis, modification, action, as well as inactivation. Now, this table also shows some of these differences between the two. Okay, so I wish you the best of luck and good luck. Allah ma'akum.